Hi, I'm Aryan Kamal, second year PhD student from Zog Group at EMBL. I'm starting with a little background. Since most of the genetic variation are in the non-coding regulatory regions and the effect of the environmental influences are very hard to measure, it's very important to use a predictive regulatory model. For this, we propose a framework for building a unified, unbiased regulatory network using cell type and condition-specific edges, and also using a universal evaluation strategy. First, the GRN reconstruction. You can see in the left panel, the network has three type of nodes: transcription factors, regulatory elements, and genes. They are connected by two types of connection, TF to enhancer and enhancer to genes. The key point of our approach is that we integrate these two type of connection, which inferred from multiple complementary methods and construct an unbiased unified regulatory network. The network is based on the data from a cohort of the paired ataxic RNA-seq samples of at least 20 individuals. In the next part, we suggest a novel evaluation pipeline on predicting cell type specific expression response to perturbation using gene regulatory networks. Briefly, we train a random forest model to learn the coefficient for each transcription factor in predicting differential expressed genes for all genes. In the last part, we are using the prediction model and not only using it to evaluate the performance of our gene regulatory network, but using it as a benchmarking tool to compare our enhancer gene regulatory network with other GRNs. Here, I briefly explain how we reconstruct the gene regulatory network. We use the HOCO-MOCO version 11 and search for the transcription factor binding site within ataxic peaks. Then we using the TF expression, we compute the correlation between transcription factor and each ataxic peak across the individual. We also compute the FDR for this correlation, and based on a specific threshold for that FDR, we filter these connections. Next, we connect peak to gene based on their distance and their expression correlation. With these two steps, we first connecting transcription factor to enhancers, and then we are connecting enhancer to genes, and directly we are connecting transcription factor to genes. In the evaluation pipeline, we are using a constructed GRN and RNA-seq data from two different conditions. First, we calculate differential expression value, then we construct the relation matrix based on the TF gene links from the network. Next, we apply a random forest regression on actual and randomized relation matrix to predict differential expression values, and we evaluate the GRN based on its performance compared to the randomized network. Here is some part of our result. In the left panel, we tested our reconstructed method using 42 paired RNA and ataxic samples for naive macrophages. As we can see, we have a network with around 110 transcription factor and 1600 genes with 2400 connections. In the part B, we wanted to see the histogram for the number of enhancer connection versus distance to the gene. And as we expected, we have more connection when we are closer to the gene. We have a R package for reconstructing the network with the complete vignette and the full workflow example that you are welcome to use it with this link here in the footnote or just scanning this QR code. We also developed a web server to explore and visualize our enhancer gene regulatory network. It is also available with the URL in the footnote or this QR code. In the right panel, we have the result for the evaluation pipeline. In A, you can see R-square for predicting differential expression between the naive macrophages and 5 hours after infected with the Salmonella using real and random network, and it shows the actual network performing much better. And in the next scatter plots, it shows a clear regression line between the predicted log-fold change and the true log-fold change. And you can see in the right scatter plot, there is a very few points with a significant log-fold change that we couldn't predict its directionality. Our next result is comparison between the network using only promoter links and the network containing enhancer links. And it shows how much enhancer links are important. Next, we investigate more on the cell type specificity of enhancer GRN, and thanks to some of my colleagues, we reconstruct the network from 5 different cell types containing naive T cell, iPSC, AML, macrophages, and brain. We tested if we can predict differential expression value for macrophages with using these networks, and as you can see, only macrophages network is able to predict differential expression in macrophages. We also compared our network with the two well-known existing network, Dorothea and T-ROST, and which both of our enhancer GRN and Dorothea perform very well. And for the final application, we used LDSC algorithm to find the connection between our enhancer GRN and GWAS trait, and we found some very interesting result when we used the most important transcription factor from macrophages, and for example, unipolar depression. Which the relation with the macrophages have been shown by several studies. And for the summary, we have a gene regulatory network that uses both RNA-seq and chromatin accessibility data and reconstruct the TF2 enhancer to gene connection. And then we introduce an evaluation algorithm and call the network predictive if we can predict perturbation in the data set using our enhancer GRN. And then we use that to optimize our network parameter and compare it with the other existing networks. And at the end, we showed our network is indeed cell type specific, and we used important transcription factor in a specific cell type to find the connection to GWAS traits. So by capturing enhancer and using the prediction model, we can understand more about the specific cell types and their gene regulatory network, and moving from a descriptive gene regulatory network to a predictive enhancer regulatory network. With this, I would like to thank you for listening.